Okay, folks, how's it going? So I just saw somebody commented about the iClicker extra question. Yeah, there were a couple of emails that I got from that. I'm sorry, I didn't reply to those actually. I just kind of got busy and then forgot about them, I'm afraid. I'll go back into the iClicker for Tuesdays and try and resolve the issue with that. There are like 5 extra iClickers that are using our drops that weren't actually part of the class. Okay, I'll have a look at the Tuesday iClicker, okay? Because I remember I clicked it, I opened it at one point and I meant to be closing a question, so there was an extra question in there, and I will do that. I'm not turning on the iClicker right now, okay? So just in case you're expecting it to come on. Tuesday iClicker note put up for myself. Okay. Let's see, are we using iClicker today? Are we using iClicker today? Not right this very second. I'll see how it goes, okay? Professor, I just had a quick question pertaining to the last lecture. So are tetrapods lobe-finned fish? Yeah, yes, they are part of that clade. They evolved from the lobe-finned fish lineage, okay? So we're within that group of the Sarcopterygiae. The mammals and vertebrates are within, well, not the vertebrates, they're vertebrates, but mammals are within, and the amniotes are within that group of the Sarcopterygiae, which are the lobe-finned fish, okay? Okay, righty-ho. So what I'm going to do today is finish off the lecture from last time, which was the Deuterostomes 2 part, chapter 31. And we'd got this far, okay? Where's my pointer? There it is. Are we skipping slides? No, we actually, oh yes, we might be. Yes, yeah, hang about. Yeah, good point. Let me just try and get my mouse. There it goes. Okay, to where it belongs. Yes, we were. We skipped a bunch of slides, right? I was just flicking through them myself before the lecture. Okay, so we were here basically. We had got to this point where we talked about the amniotes, okay? Now, if you remember, that was the step that came kind of after the amphibians, tetrapods moving onto land. And the amphibians were kind of, are constrained to kind of wet habitats, especially for reproduction, okay? Because the eggs are not adapted in most circumstances to be able to develop outside of water. Amniotes developed this egg that we talked about with the 4 membranes that resists water loss from the embryo, but allows gas exchange. So that allows development to occur away from water. Now, soon after the amniotes arose, they were split into 2 groups. We spoke about the reptiles last time. That includes the living groups of the lepidosaurs, so snakes and lizards, plus 1 live species of tuatara in New Zealand, turtles, crocodilians, and then living today, the birds, the last surviving sort of group of the dinosaurs, which mostly went extinct about 66 million years ago with a large impact on Earth from an asteroid or something similar. So we talked about those reptiles. Now we're going to talk about the mammals, okay? So that's this other group of amniotes that split from the reptiles early after the amniotes arose. Okay, so the first mammals arose about 225 million years ago, and they were small nocturnal insectivores, so eating insects. Because remember, the insects moved onto land way before any of the vertebrates did. But they remained in that kind of condition until most of the dinosaurs went extinct, except for the bird lineage about 66 million years ago. And that opened a whole sort of new world to these now that that competition from the dinosaurs had been removed. So today there are about 5,700 living species of mammals, and they're characterized by 4 key characteristics. The first of these is the sweat glands. Okay, So we have sweat glands on our skin. They secrete sweat onto the surface of the skin, and as that evaporates, that cools the body. I believe it is the last class of slides we missed. Don't quite understand your point, Jade. I believe it's from the last class, the slides we missed. This is a continuation of the last class, and I think we got on to, I looked at the video, and we started on the mammal video slide, okay? So, the second key characteristic of the mammals is mammary glands, after which the group is named. So that is glands in the skin that can secrete milk for nurturing offspring. And in its earliest form, that arose as just patches of skin that had secretions that then the juveniles could drink. In more derived forms, that has sort of specialized into, you know, the nipple that has concentrated, specialized mammary glands just for that area. A third feature is hair, and for insulation and for protection. And the fourth is a four-chambered heart. So we talked amongst the reptiles, or some of the reptiles, about having a three-chamber heart, how the heart evolved from thickening of a blood vessel on the ventral side of amniotes and before, and became then more specialized. So muscular, but then division into separate chambers that could sort of pump and segregate blood in a more efficient manner. The four-chambered heart of mammals, the benefit of that is it allows us to differentiate the circulation that goes to our lungs for gas exchange from the circulation that goes to the rest of our body for providing oxygen and removing carbon dioxide. Okay, so it's a more efficient way of providing the body. So mammary glands were before were just random spots in the skin instead of, yes, correct. Mammary glands were just, there was no specialized nipple. It was like patches of skin that then became more specialized over time. It's unlikely today. Not right now, we'll see maybe later on, okay? But I don't have it activated right now. So the other feature about mammals is that development of the embryo became internalized. Okay, so that involved internal fertilization of the eggs instead of external fertilization. And nearly all species, although we'll talk about some exceptions, have internal development of the embryo within the uterus. And the way that has evolved is basically that amniotic egg that we discussed, how it evolved with these four different membranes, the yolk sac, chorion, allantois, and the amnion. They now are part of the internal structure, and there are homologous features that make up our umbilical cord, and then which connects with the placenta. So you can see here, this yolk sac is present as part of the umbilical cord there. It's become much reduced in, what is the allantois response? I'm trying to remember, in the previous, some of the earlier slides, it gives their specializations, and I don't specifically remember, I'm sorry. So the key thing with this is they're homologous features as part of this. Waste removal, thank you, Isaac, you're better than I am. So, and the placenta allows the blood from the embryo to come into very close contact with the mother so they can exchange nutrients and gases across that, okay? So they can nurture the development of the embryo. So here, the placenta is the other key feature here, connects embryo to uterus wall, nutrients, gas exchange, etc., okay? So here's a simplified phylogeny of the amniotes with the reptiles that separated early on. This is the ancestral amniotes at the root of the tree here. And we're going to talk about these mammals that are split into the prototherians, the marsupials, and the eutherians, okay? And these two together are considered to be therians, okay? So let's go through each of those. So the therians are the basal group amongst these, and as the sister group to the therians. Okay. Now, the only living species of these prototherians today belong to the group the monotremes. That translates from Greek as meaning one whole, because they still have a fairly ancestral condition where they have a cloaca, which is a single opening that before that both the anus and the urogenital tract enters into an area, and then there's a single opening from that cloaca, as in the reptiles. I also mentioned that with the evolution of mammary glands, initially there was no nipple, and they retain this basal feature of just having skin patches where those mammary glands occur. And the living species of those include the platypus. There's one species, lives in Australia and Tasmania, you know, with this distinctive, really weird bill. And then the echidnas. There are 4 living species of those that live in Australia and New Guinea. So these are egg-laying mammals. Okay, so they haven't fully internalized that development of the embryo yet. The next group beyond that, amongst the therians, are the marsupials. So the marsupials, they have now a simple placenta, so internal development, but a very short gestation time. So their placenta is not nearly as developed as in ourselves, for example. And it's a yolk sac placenta, it's called. It's made out of the yolk sac, and it supplies yolk nutrients to the uterus, but no direct exchange between the mother's blood supply. Okay, and so because of that, that's what leads to the short gestation time. So they're born at a very, very early stage of development, and then they crawl to this ventral pouch, shown on this wallaby, I think it is, and attached to a nipple. So now these do have a specialized nipple, and then they develop eventually into this little joey here, before they then become independent. So they're tiny at birth, and they occur in the Americas and Australia. So particularly South America, there's only one species in North America, the Virginia possum. So, you know, what we see around here. But it involves things like kangaroos, wallabies, koalas, etc., and Tasmanian devil in Tasmania. And then the third group is these eutherians, the so-called placental mammals. But it's in inverted commas because you know, these do have a sort of basic type of a placenta, but this is a fully developed placenta, as I showed in that earlier diagram in the mammals, that allows exchange of gases, nutrients, and waste products between the embryonic blood supply and the mother's blood supply. Okay, so about 5,300 living species and 20 major groups of living species amongst these. So this is a table from the book showing these different groups of eutherian mammals. And so the point of this is not for you to memorize. You don't have to memorize this entire table. Is to highlight a few points. And one of these is that amongst the most speciose or diverse groups of these eutherian mammals is at the top rodents. So the rodents are characterized by special types of teeth that are designed for gnawing through things like wood and so on. So that's, you know, I said there were how many? 5,300 species. So about half of those are rodents. Now, the second most specious group is, perhaps surprisingly, is the bats. So here is a group of mammals that evolved flight. So remember, these vertebrates all originally evolved in the ocean, and then invaded land, especially with the evolution of the amniotic egg. And now some of these are evolving flight. And so this is one of the reasons why this is probably a speciation group, is this allows high dispersal, so they can go to all sorts of new different habitats. It's akin to the way that when insects invaded land and had flight and then were able to spread, in their instance, you know, being the first animals spreading across. So the other major groups amongst the most speciose are the even-toed hoofed mammals, things like deer and sheep and goats, etc. Shrews, primates, we'll talk about those at the end, and carnivores, things like wolves and dogs and bears, etc. Okay. Now, some of these mammals actually, you know, so these animals have originated from the ocean, invaded land, and now some of them have evolved to go back into water again. And this has happened independently in separate groups. And so whales and dolphins, for example, the cetaceans, it was for a long time, you know, which group they had originated from was quite a mystery, but genetics shows us now that they're closely related to the even-toed hoofed mammals, and in particular their closest land relative is the hippopotamus. Okay, and they've evolved to go back into water, as have the pinnipeds, the seals, the sea lions and walruses, and the manatees and dugongs, and amongst here somewhere should be the otters as well, which have gone back into both freshwater and seawater as well. Now, if you look at the phylogeny of the eutherian or placental mammals, there's some sort of grouping that occurs by land mass. This slide here is showing you the fact that due to continental drift, the continents and the shape of land masses around the Earth has moved and changed over time. So when mammals first arose about 225 million years ago, there was this big single landmass, Pangaea. Okay, but over time, it broke apart. So here there was a northern continent, Laurasia, and a southern one, Gondwanaland. And then around about the time that the dinosaurs went extinct, about sort of 66 million years ago, landmass was like this. So South America is much closer to Africa than it is now, but they were splitting apart. So this is what the mammals first started really sort of diversifying in. And if you look at the genetic phylogeny of modern day animals, there's some grouping that occurs within that. So this column here is showing when whereabouts the earliest fossils of each of these groups was found. And you can see that there's a sort of grouping amongst African mammals, modern day mammals, South Americans, etc. This Laurasian, that northern continent, they group together. So what happened was that certain groups got separated on these landmasses as the landmasses moved apart, and then evolved new groups independently within those. And then certain groups have perhaps come back into contact with one another, or there's been movement somehow between groups. Then there's been some sort of mixing. So for example, South America and North America, they joined back together with one another at the Panama Isthmus about 2.8 million years ago or so. And so armadillos now occur within both North America and South America, even though they evolved initially in South America. So there has been exchange amongst these. What we're going to talk about now is just one of these groups, the primates. And so we're going to talk about those because, you know, they're of interest to us, seeing as we ultimately belong to that group. So let's talk about the primates and some of their key features. Okay, so originally, when they arose, they were arboreal. That means they lived in trees. And so they're adapted for those kind of conditions. They have limbs that are grasping limbs, so they could grab onto branches, etc. An opposable thumb. So an opposable thumb means that you can, as in this picture here, we can put our thumb across onto the far finger like that, meaning it's opposable, which other types of animals can't do necessarily. And then they have overlapping binocular vision. This isn't purely unique to the primates, but the advantage of this is primates are eutherians. Yes, they are. Okay. Yeah, so eutherians, these are all eutherians in this group here, of which primates are those, so placental mammals, right? So the advantage of binocular vision is it gives depth perception. So if you're sort of, swinging through the trees and jumping, etc., it gives you that depth perception that you need in order to do that without falling, basically. Okay. So let's look at the, what we're going to do is follow different lineages of primates that ultimately lead to the evolution of modern day humans. So primates split into two major groups about 90 million years ago. Okay, so we talked about the mammals first arose about 225 million years ago. Now, soon after they arose, they split into two groups, and the first of these is referred to as the wet-nosed primates, and this contains the lemurs and the lorises. Okay, so today the lemurs, they occur in Madagascar, and lorises occur in equatorial Africa and Asia. And they're arboreal. Their ancestral condition is that they are nocturnal. Except in Madagascar, they have radiated since that time into some other forms as well. There are some forms that are diurnal, so they go out in the daytime or are active in the daytime, and some forms that are terrestrial as well. So they've radiated away from that ancestral condition in some instances. Yeah, so these wet-nosed primates, they also have smaller brains and larger olfactory lobes, so for smelling, basically, and very well developed eyes as part of that nocturnal adaptation. So the other major group are the so-called dry-nosed primates. Now, they split early on into the tarsiers, which is a minor group. I think there's maybe one or two. There's a few species of those. But then the major splits occurred earlier on, around about 40 million years ago, into the New World monkeys. So that means Central and South America, and the Old World monkeys in Africa and Asia. Now, it's thought that these New World monkeys probably originally arose in Africa and then were able to move to South and Central America, either possibly because there was some land bridge, or more likely it's thought by some drifting or rafting, etc., which animals are known to move between areas in that manner. And that time, the continents were much closer together, which would have facilitated that. So these New World monkeys, they're arboreal, so they live in trees, and they're characterized by having this prehensile tail. So as in this monkey here, that's able to wrap its tail around a branch and hang on to them, as opposed to the Old World monkeys that are not able to do that. So that's the distinction between the groups. So amongst the Old World, they are arboreal, but there are also some that are terrestrial, okay, so living on land. And as I mentioned, their tail is not prehensile. And also within those, there is a split into the apes, so the Old World monkeys, and then the apes that come off that involve the gibbons, orangutans, and then the African apes, and ultimately humans. So the apes split from the Old World monkeys. And they live in Asia. It includes the gibbons and the orangutans, 3 species and 18 species respectively. And then in Africa, 2 species of gorillas, 2 species of chimpanzees, and humans ultimately that then went on to colonize the world. Okay, and the fossil apes, there's also some fossil apes from Europe around about 7 million years ago. And sometimes people refer to as the great apes. The great apes includes the African apes, but it also includes the orangutans as well. So as part of this, did I skip? No. Okay, so we're going to move on to evolution of humans and the group Homo, okay, that we belong to. And as part of that evolutionary change, we adopted a bipedal posture. Okay, so we're walking on 2 legs, and some of the advantages of those was that it frees the forelimbs, and now the forelimbs can be used for other purposes such as manipulating or carrying things or weapon use, etc. Also, it elevates the eyes. So these are terrestrial animals, and so elevating the eyes means that you can see things further away, such as spotting predators, for example, or if you're hunting, you can see prey. And the other thing is, it's energetically more economical than moving around on land using all 4s in the way that the other apes were doing. So this is a phylogeny of how that sort of, I know snakes see heat. What is? I know snakes see heat. I don't know what it's called, and it's not so relevant to this slide, I guess. Infrared vision, perhaps. Yes, okay, Mark. So this is a lineage of human evolution, basically. So we had these bipedal adaptations in these early ancestors. And the point of this slide, or part of the point of this slide, is to illustrate that as this is a kind of a simplified view of the evolution of ultimately of the genus Homo, and it's way more complex than this, and still not that well understood. But there were 3 main groups shown on this slide: the Australopithecus, which was an early group that separated, followed by the Paranthropus, the second genus right here, and then Homo down at the bottom. And so this Australopithecus, the most famous specimen of this is the fossil Lucy, which was an almost complete fossil found in Eastern Africa, I think in the 1970s. That is about 3.5 million years old. But it overlapped in time before they ultimately became extinct around about this period here. And it overlapped with this group. So these different groups were actually in coexistence with one another. And then interestingly, this group, the Paranthropus, overlapped in time with Homo as well. So this group, the Paranthropus, overlapped with both of these major groups. Not with us, but with our ancestors. Now down here, once you get into the Homo genus, there's different colors here, and the light blue indicates those groups that radiated and expanded and moved out of Africa into other areas. So we are not the only species of Homo that has moved out of Africa in the past. Homo florensis, which occurred in Indonesia, moved out way before we did. So when was this? Like 1.5 million years ago. But ultimately became extinct. Same with Homo erectus, which was more prevalent in the sort of Europe and Asia regions. Okay, so here's Homo coexist in Africa, Asia, and Europe until recently, yeah. And then, so we're down here, and expansion into the New World 12 to 20,000 years ago. But then expansion from Africa about 60 to 70,000 years of growth, okay, the group that we're in, Homo sapiens. But earlier to that, along a similar thread, humans are a relatively new species. So this has taken it back to, what's that, about 500,000 years ago, if I look at that separation there from Neanderthals versus Homo sapiens, okay? Now, Neanderthals also moved into, out of Africa, and they did so, way before we did, basically. But when we moved out of Africa, we overlapped in time with that. So there are instances in, caves in southern parts of Europe, I think Spain and perhaps Gibraltar, where there are fossils of Neanderthals coexisting with modern day humans. And there's evidence, there's strong evidence that there was exchange, breeding between them. So if you go on, 23andMe or, these companies now, what caused them to go extinct? Apparently we did. So it's thought. But we have in us, about I've done 23andMe and I can't remember my percent, but it's a small percentage of our genome actually comprises Neanderthals. And there was another group as well. It's not shown on this diagram, but it's in the notes below, the Denisovians. They were discovered in 2010, I believe. And actually, modern day humans have some genetic component from those Denisovians as well. Another group that moved out of Africa where there was interbreeding. I think they moved out at an earlier stage. And modern day humans that have that, it's more prevalent in the genome in sort of natives of Australia and New Guinea, I think.
Where did the New World monkeys, where did they figure into this diagram? Were they happening when Homo erectus was expanding out of Africa, or? No,
so that was at a way earlier stage, basically.
So those New World monkeys were already there when our Homo species expanded out?
Yeah, so this split was, like 90 million years ago. Primates split into two major groups 90 million years ago. Okay, into this, so this is into the wet nose and dry nose, but amongst this was occurring about 40 million years ago, okay? New World monkeys, Old World monkeys, okay? That's the split you're asking about, right? And then what we're talking about here is, 4 million years ago from this point onwards. So at a much, much more recent stage, okay?
Thank you.
So as part of this evolution of Homo, a general sort of change that has occurred over that time is an increase in brain size. And this occurred simultaneously with a decrease in jaw muscle size. And the reason for that is not completely clear, but one of the possible explanations is there's some developmental link between that. If you can't do one necessarily without compromising on the other. But I don't know the specific details of that, but that's one of the hypotheses on that. Is that still relevant among humans? Which bit, the brain size part, or don't know, I'm not sure. Jaw and brain. Well, we have a smaller jaw than these much earlier forms, okay? And that was also characterized by some of these early forms. It's not shown so well, but Homo erectus, for example, I think, had a very, very prominent crest on the top of the skull, and that was for allows really big muscle attachment that drives the jaws. So, okay, and what was this brain size related to? Well, the hypotheses related to that is that it's associated with more complex social life that developed, and things like rituals. So, evidence of burials, ultimately use of clothes and tools, etc. In more derived or recent forms, art, in cave paintings, language and cultural transmission. So towards the movement towards a sort of more society, basically. Those are the sort of general hypotheses of what that's related to. And this slide, this final slide really is just showing how recent some of this, you know, movement and radiation of humans around the world has occurred. This is a diagram that shows, so this is in thousands of years, and basically the redder parts indicate later, you know, earlier movement, and the bluer parts indicate more recent movement. So from this area in Eastern Africa, radiation of about, you know, movement about 200,000 years ago, movement out of Africa here about 70,000 years ago, and then ultimately movement around the globe. These blue dotted areas here indicate areas that were covered in the last ice age about 20,000 years ago. That might have restricted some movement in some ways, but in other ways it could have benefited movement because the sea level was much, much lower. So there were, because all the water was locked up in ice mass. So there was more land bridges present as well. And if you look at like New Zealand, right, 700 years ago, I mean, that's like nothing. There are buildings, if you go around, you know, Spain and Italy and Britain, etc., that are older than this, when the Maoris first moved to New Zealand, and Hawaii, you know, 1200, 1600 years ago. So recent compared to the grand time frame of what we've been talking about in general. So it's kind of interesting to look at these movement patterns and the timing of them. So that's the end of the deuterostomes, and I just want to sort of zoom out again as well, because it's really also the end of a whole series of lectures that we've had since the beginning of the semester, really, that have taken you from the beginning of life on Earth, following down these phylogenies. Right now, you know, we've gone down to the sort of tip of the primates, basically, but we could have gone down to the tip of, you know, all these other potential groups. And so if we sort of zoom back at where we've come, yeah, so here, you know, zooming back out again, a reminder of what we've covered. So since I started giving the lectures, I started talking about the animals that evolved about 700 to 800 million years ago. We've just come, you know, we're just talking about mammals, which ultimately were part of the chordates that belong to the deuterostomes. We talked about the protostomes. And then these other more basal animals, the monoblastic and diploblastic animals. So I took over at this point in time, okay? And before that, Dr. Spagnolo, so here are the animals, you know, he started off with when life first began on Earth, about, you know, 3.7 to 4.4 billion years ago. So, you know, soon after the Earth came into origination, basically. Eukaryotes first developed about 2.7 billion years ago. And so we've, you know, just been covering the tip of the iceberg, basically. So it's interesting to sort of, or it's useful to just put all of this into the grand perspective of life, okay, and everything that we've managed to cover. Okay, and as usual, I put in some summary slides on here that I'll leave you to go through, but the main summaries of these is to remind you of the major phylogenies that we've passed through, okay, to get to what we've just talked about today, okay. It's useful to think about those in their perspective. Okay, so how are we doing? It is now 12:53. I am going to close this slideshow down and move on to slideshow next, okay. So let me just switch these screens and get ourselves sorted out here. Okay, so now for something completely different. What we're going to do for the rest of the semester is we've covered, you know, a huge aspect of life on Earth, basically. In this lecture, I'm going to talk about animal, and this will continue to the next lecture as well, animal behavior, okay? And then I'm just looking at my schedule here. Beyond that, we have 6 lectures that are covering general aspects of ecology. So we're moving on to a sort of the next step, basically, okay? So this is chapter 51, talking about animal behavior. Get my pointer going again. So, you know, if we look at animals all around us, we can see behavior occurring. Animals behave, we behave, etc. And some of the aspects that we're going to talk about in the lecture are how are behaviors controlled, what, you know, makes them occur basically. But also towards the end of the lecture, we're going to talk about why do behaviors occur? Because behaviors, presumably, one aspect that we're going to get out of the lecture is that behavior can have a strong genetic component to it. And we know that, you know, if something is controlled by genetics, then it's subject to evolution, because if you have some genetic components that confer a trait that is more advantageous to the animal in terms of its reproductive ability, then those genes are more likely to be passed on to the next generation. That is evolution, okay, the change in allele frequency over time. So why do some of these behaviors occur? What is the advantage or disadvantage of having different types of behaviors? So some of the earlier studies on behavior that really sort of caught attention were studies on so-called conditioned reflexes. And one of the most famous examples of this that you probably heard of is Pavlov's dogs. Psych. You mean that psychology? Is that what you're saying? Perhaps. So yes. So so Pavlov's dogs that many people heard about, he was interested in this sort of salivating behavior of them. He referred to the conditioned reflex as linking a sound stimulus to receiving food. Okay, so he noticed during his studies that he had these dogs, and when his assistant, who was the person who fed them, would come in and feed them, before they'd been fed, on sight of the assistants, these dogs would start salivating in anticipation of the fact that they were going to receive food. But they also, you know, salivate if they just see food, and he referred to that as the unconditioned stimulus. So it's something that's autonomic or automatic, effectively, within the dog. It's an involuntary response, and it doesn't depend upon necessarily previous experience. It's something that that they're born with, learning by association. Yes. But what he was able to do was then he did some experiments where he used a metronome to make a sound. Okay, and so he conditioned these dogs that when they were about to be fed, he would sort of, first of all, play them this metronome and condition them so that they associated the metronome with the food in the same way that these dogs were associating the assistant's arrival with the food, and they would start salivating. And then ultimately, he could then just play the metronome without any food, and you had this conditioned reflex, okay? So the reflex being the fact that they would just salivate in response to this metronome. So he received the Nobel Prize as, you know, this work was part of what he received the Nobel Prize for in physiology and medicine in 1904. And in fact, in the sort of history of studying behavior, there's a number of different Nobel Prizes that have been won by people, most recent being in 2017, and we'll discuss those as we go through the lecture. Another one of these early studies on conditioned reflexes was by Skinner, and he was interested in the operant conditioning. So that was so named because animals learn to perform an operation on its environment to receive a reward. I've got in the notes section there from a web page that I took. Okay, and he studied rats, and so this was like his became known as a Skinner box, this apparatus I'm showing here, which is like in a way a little bit like a mini torture chamber in some respects. But he would put a rat into this box, and then one of the experiments that he did was that this box has a little lever here. And in one of the experiments, if the rat moved around and accidentally knocked this lever, then it would release a little bit of food that would come in, and the rat would eat it. And over time, the rat came to learn the fact that if it pushed this lever, then it could receive food. So it would do that when it was hungry. And you can do like equivalent experiments to be done on fish, for example. So fish can also learn this type of behavior as well, in a tank, obviously, not in a Skinner box. Another experiment that he did was instead of giving a food reward, this little floor here, he could pass an electric current through it. Birds too, yes, birds absolutely can do that. So it's not just confined to mammals, for example. So what he would do is he'd pass an electric current, presumably a mild one, I'm not quite sure, through this box, and the rat would run around in an uncomfortable manner, and it would knock into this lever, and it would turn off the electricity. And in the same way, the rat could learn that it could, if this became electrified, it could run over and switch off this switch. And then he actually developed a whole bunch of more complicated experiments involving multiple rats and things like that led on from these types of studies. So these early studies that I've just talked about came collectively known to be termed as behaviorism. So these were behavioral studies stimulated particularly by the work of Pavlos and Skinner, but also other workers as well. And the focus of these were that they were experiments on a few sort of lab or convenient species under highly controlled laboratory conditions. So you could put them, like as you might do a great experiment like these guys did, you'd have a highly controlled condition with something that you could keep in a lab and sort of manipulate or experiment to sort of test your hypotheses, basically. Some of the questions that arose from this from other people, though, was what about animals in their natural behavior? You can sort of tease some of these things apart, but what does this mean in the real world, basically? And this led to the development of a group of studies generally called ethology, okay, which was primarily interested in animal behavior in the context of their natural environments rather than under just these specific laboratory controlled conditions. And was also interested about how are some of these behaviors adaptive to the natural environments? What's the advantage or disadvantage of some of them? How did these things evolve? And it involved a broad range of species, not just one or two convenient laboratory species, basically. Like I said, moving out into the real world to see what the relevance of all this is. Some of the early leaders in the field of ethology were these three people here. Okay, now I mentioned that there were several Nobel Prizes that have been won in the field of behavioral studies. These three people shared the Nobel Prize in 1973, again in physiology and medicine. And these involved, so there was Carl von Frisch, and he was famous for, one of the things he was famous for was deciphering this so-called waggle dance of honeybees. So when bees go out and look for food, they return to the hive, and then they have a specific dance that they can do that can confer to other members of the hive of like which direction with respect to the sun they have to fly and how far they have to fly to where that food source is, so that others can go and get food source. So he was able to decipher that language effectively of that waggle dance. And then they led to studies on other species to show that they do the same thing, but they have a different language. They confer in a different way. Another famous leader in the field was Konrad Lorenz. Okay, so he studied the strong bond between parent and bird offspring, particularly imprinting that we'll talk about, and its development during an early critical period following birth. And so he would get various birds to imprint on him, like these two, for example, here, that would follow him around thinking that they were his mother, basically. And then Nico Tinbergen, he was a Dutch scientist. He worked at Oxford University. He studied inborn patterns of behavior or instincts in animals and their adaptive and evolutionary aspects. So we'll talk about different components of some of these people's work. So in ethology, The interest is in instinctive behaviors referred to as fixed action patterns, and particularly the examination of how there's some genetic control of some of those types of behaviors, because that genetic control indicates a mechanism of evolution, basically, or a necessary part for these behaviors to evolve. So behavior thought to be genetic if it occurs without learning, and if it's stereotypical, that means it's performed in the same way each time and cannot be modified by learning. That was the sort of thinking behind this. But how do we actually know that something is genetic? So when these studies first arose, you know, this was not in an era where you could go out and sequence something's genome or anything like that. You know, let's think when these guys were studying, you know, back in, they were sort of really active in the 50s, and in particular, you know, DNA was even hardly sort of discovered yet, basically, by that point. But there are some ways that you can decipher the fact that behaviors are under genetic control, and one of the methods used was using these so-called deprivation experiments. Sorry, that's my cat in the background that you might be able to hear. So early ethologists placed considerable emphasis on these deprivation experiments. Okay, so what does that mean? So an example of this is with spiders. Now, I think I showed this diagram actually in an earlier lecture. It's not from the book, but it shows the fact that different types of spiders have characteristic different types of webs that they build. Okay, some of them build, you know, webs on the ground as part of holes, etc. Some of them have your classic thing like this. Others, like this one here, you know, are like little nets they hold, and their limbs for hunting, etc. And so the types of and shape of web that you get is characteristic of different species. And what they did was they were able to raise juvenile spiders. Scorpion is a spider. Scorpion is an arachnid, okay, but distinct from an actual spider. So juveniles raised, and so what they did was that you could raise a juvenile, and so it's never exposed in any other way, deprived of other spiders and of other webs, etc. There's no way it can learn it, and then just see what happens, and it builds that web. Okay, so that was an indication through these deprivation experiments that these were that there's a genetic control to this. This was some of the earliest evidence of this. Okay, I'm also going to talk about some of the work of Tinbergen, and who was one of those 3 people who got a Nobel Prize. And one of the things he was interested in was a so-called releaser, a specific stimulus that elicits a fixed action pattern or an innate behavior. Okay, and it built the species specific. It built the species specific web. Yes, it built. Going back to this deprivation experiment, you raise a spider in isolation, let it build a web, and it builds just a normal web, basically. It's never, you know, so indicating it's not like learned it or anything like that from something else. So in Tinbergen's experiment, he was interested in this innate behavior of chick gulls, seagulls, and so experimental role in chick begging behavior. Yeah, so when an adult gull comes back to the nest, these chicks will start to beg for food. But you don't necessarily want to be doing that all the time for the chick, because you only want to beg when there's food around. You don't want to be drawing in predators and etc. if there's nothing else around there, or wasting energy. And what he was able to show was that this little red dot here that occurs on the lower beak of a gull is a releaser. It can trigger the behavior of the begging chick. And he did a whole bunch of experiments where he had model gull heads, basically, that either looked exactly like a gull, or he had one that had the shape of a gull's head but no red dot, or something that didn't look very much like a natural gull's head, but it did have a red dot, and this could elicit a strong response similar to an actual gull, but without the red dot, you wouldn't get that. And then he manipulated things like the shape of the head of the model and the color of the head, etc. And basically, the story was that as long as you had a red dot, you were in good shape. If you didn't have that red dot, then you got a weak response, even if you had a realistic-looking gull head. So this was a releaser of this innate behavior. So we're talking about the genetics of behavior. Another way that you can understand that behavior is controlled by genetics is through interbreeding. Okay, and Lorentz did some experiments on this on dabbling ducks. So different species of ducks, the males have a characteristic sort of little dance that they go through when they're courting, basically. So in this example here, you know, it starts off with one type, it's a tail shake, followed by a head flick, followed by a tail shake, etc. And they go through this series sort of stereotypical manner of that species. But you can go to another species that might be fairly closely related, and they may have a courtship dance they go through, but there's a difference that occurs. So he did breeding between these types of ducks, and then looked at what the courtship dance was in the offspring, and found that basically you get a mixture of what they came from. So some hybrids, also between other species. Okay, yes. Yeah, so in the notes, I'm just reading what it says on the notes part here, was that in those hybrids that had the mixed behavior, if you try to expose them to a receptive or sexually mature female, they weren't successful in, or were generally unsuccessful in being able to mate with that female, because the female was selecting specifically for the type of dance that is characteristic of the species. So there's some sexual selection that goes on that reinforces that behavioral trait within males of the conspecifics. So some of these early ethology studies paved the way for modern behavioral studies. So ethology, if you remember, is the study of behavior of animals in their natural context. And it led to questions such as, what are the proximate causes of behavior? And by proximate, I mean, what are the actual underlying mechanisms that are involved in controlling that behavior? And how do they develop or perhaps change with developmental time? So what experience might be needed, and what are the ontogenetic changes? So ontogenetic means changing with development stage. And also, we'll go on to talk about what are some of the ultimate causes of behavior. Okay, so what is the function? What are the fitness benefits? So fitness is effectively referring to how effectively you can pass on your genes to the next generation. And how can some of these behaviors evolve if they are under genetic control? What are the sort of evolutionary forces that are shaping those behaviors? And a lot of the study of this has become sort of facilitated over time with increases in technology and understanding of genetics, etc. So one of the studies of this, looking at the genetic control, was using mutants of Drosophila fruit flies. Okay, let me just check the time here, see how we're doing. (1:14). Okay, we've got 15 minutes. Okay, so Drosophila, they have a built-in, like pretty much all animals have a built-in circadian rhythm. So what is a circadian rhythm? That basically, circa means around or about, and the dn refers to a day cycle. So this is it means about a day's cycle. So many animals, or most animals, have a built-in circadian rhythm, whereby activity levels vary during the time of the day. Like we go to sleep at nighttime normally, and we normally, do stuff during the daytime. And those circadian patterns can persist even if you take away light cues and things like that in some manner. So what they did in this study was they generated mutants and found that a mutation, ultimately they found this out, it took a while to find out, but a mutation, a single gene called period, can disrupt the circadian rhythms of Drosophila. So this was first discovered or first reported in 1971. And this is showing this bottom diagram. I'll just explain what these bottom diagrams mean. So in panel A here, you can see this represents a 24-hour time period here. And if you're moving along a line, this is time. And when you've got these little black areas, that indicates a period of high activity. So moving along this top line, that's 1 day, and it's active here. Now we're into the second day where there's low activity, and in the second day, high activity. Now, the way that these periodograms work is that this second day here is now repeated again on this second line. And then, so we're going to day 1, day 2, day 2, day 3, day 3, day 4, etc. Okay, and you can see that some periods of the day are periods of high activity, and it's occurring approximately over a 1-day cycle. Okay, a circadian rhythm. They exposed a bunch of fruit flies to a chemical mutagens that led to high rates of mutation and generated lots and lots of offspring. And then they screened lots of these offspring to look at their activity cycles, and they found three examples of disruption. So in this section here, panel B, referred to as a rhythmic mutant, you can see that they basically don't really have much of a discernible pattern, certainly not over the course of a day. There's some high activity here, then it stops a bit, and then it starts and stops, etc. The pattern is not like that A. Okay. Then there was a short rhythm mutant. So they have some kind of a circadian rhythm built in there, but the period of being active is a shorter period of activity compared to the wild type one. And then there's the long rhythm one, okay? Long periods of activity with short periods of inactivity. So this indicates, and you could breed these lines indicating there was genetics for this. And it wasn't until 1984, so 13 years later, they were able to sequence this gene and actually find the mutations, specific mutations in a single gene that were causing this change in the circadian rhythm. So these patterns can also be revealed by so-called knockout experiments, which I think maybe Dr. Spagnuolo might have talked about, this knockout mechanism, but it's a technique or a group of techniques that can be used to go in and knock out a single gene within an animal, basically. So knockout experiments can reveal genetics of behavior or the proximate control. So with knockout experiments, a specific gene can be made inoperable, and then you screen the mutants to see what has been disrupted. An example of this was with males. So male mice, they have a single gene that codes for a pheromone receptor, and this could be knocked out. And in the wild type males, so the non-mutants basically, if you expose them to a female, they will mate with that female. But if you expose them to a male, they will start to fight with that male. Conversely, if you get the knockout, it mates with a female, or you expose it to a male and it mates with a male as well, because it can't distinguish between one and the other. So the conclusion from this experiment was that the genes controlling the pheromone receptor development is needed in order to discriminate the sex, and that makes a difference in the behavior of that male mouse. But often, we hear in the news about the scientists have discovered the gene for something. But in reality, that's generally not the case, because genes act together, or often in cascades. So there's like a knock-on effect between one gene and another, and one influencing the other, etc. Okay. So mutations in a single gene can disrupt behavior, but genes typically act together in this cascade pattern. So this is the contrast between the gene for something versus what's more likely is a gene controlling behavior. An example of that also comes from Drosophila, and they have a characteristic type of sexual courtship dance that they go through. That's shown in general in this bottom pattern here, where a male that's trying to mate with a female, they do some singing, it's referred to, and they tap and they stick out a wing, and they go through this little these maneuvers, basically ultimately leading to mating with a female. Now this general sex difference between male and female Drosophila is controlled by a cascade of genes together that act. There's one called sex lethal, another one called transformer, double sex, and fruitless. Okay. Now these first two, they control messenger RNA splicing. So if you remember what that is, if you have a gene and it's transcribed into messenger RNA, initially it's going to include all sorts of things like the introns of the gene, etc. So that's the introns are the bits that are not coding for protein. That's represented here by these black parts. And before that bit of messenger RNA is then translated into protein, those bits of introns are snipped out, basically, so that you just have then the messenger RNA that here is the protein. So there's modification that occurs of the RNA, and that's referred to as splicing. And and these genes here also have this red represents a stop codon. So in order for this to work, this stop codon has to be removed as well from the messenger RNA. Now the double sex controls whether you get a female or a male anatomy. And then the fruitless controls this courtship type of dance. So the way that these genes interact with one another is that in females, this gene is transcribed and spliced in a particular way. So the sex lethal controls the splicing with the transformer. Yeah, so it's produced and spliced in a characteristic way in females that then influences how transformer ultimately is spliced and the protein that produces. And then this transformer influences the splicing of double sex and of fruitless. And the way that it influences is it produces this form of messenger RNA that leads to the female morphology, and it means that you don't get the courtship dance by the females. Okay. Now, in the males, something different occurs. In males, the sex lethal and transformer are spliced differently in a way that includes this stop codon here, and it makes them non-functional. Now, if you remember in the females, that was influencing the splicing of that, which was influencing the splicing of that, etc. So that is cut out now. And instead, splicing occurs in a different manner. These are still functional proteins, but what this is showing, that this darker shade of blue here indicates that the male form of this includes these darker blue bits here that were not in the female transcribed messenger RNA. Same with this in the males, in the fruitless gene. So this now leads to the male morphology, and it leads to this behavioral difference as well. So you change this, and you're going to change that. You change this, and you're going to change that, etc. Okay, it's a cascade effect. You can have a mutation in a single gene, and it will modify behavior. But that mutation could actually have occurred in in a series of different genes that would have led to perhaps the same or a similar response. So genes can control behavior, but it's more typical that there's going to be a series of genes acting together rather than a single gene for something. Okay, conclusion, probably saying the same thing. Single mutation anywhere in a gene cascade can affect other genes, thus modifying behavior. Yes, that's kind of what I said, I guess. Okay, so I mentioned briefly earlier on that expressing, I think at the beginning of the lecture, expressing behaviors at different times can be beneficial or detrimental. And so you want to be able to express behavior at an appropriate time when it is beneficial to you. Okay, so behaviors are only needed at certain times, and expressing behavior when not needed might be risky or reduce your fitness rather than increasing your fitness. So an example of that is the begging behavior in chicks that I mentioned. If there's no parent coming back to the nest to give you food and you're just begging, 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 you're telling every predator within a radius of, you know, however far, that there's a little baby chick unguarded in the nest and you can come and eat me. At this time of year, if you walk around, you know, lakes and ponds and things like that, all the frogs are starting to sing to mate. If I go down to one of the lakes nearby us and the frogs are singing, as soon as you come near it, they all stop immediately because they've detected that I am approaching them and, you know, potential predator. So they switch that behavior off until, you know, they perceive it as being all clear. So behaviors need to be expressed at a time when they are beneficial and turned off when they are non-beneficial. So the book describes this kind of great series of experiments with these kind of cool diagrams here, basically, and I'll describe this experiment in the book here. So we're going to talk about hormones and how hormones can be part of the process that is controlling the timing of when behaviors occur, but also the timing of the hormones is affecting the behavior. So this was some experiments done on manipulation of rat hormones, demonstrating their role in behavior. So in the females, when they are receptive to mating, they have this stance here, referred to as lordosis. And the male, when they are receptive to mating, they mate basically with females that are doing this. So how is this controlled by the hormones within the rat? So in these experiments, and I'll kind of walk you through how they did this and give you a sort of overall conclusion for them. Let's see if we've got time to do this. 2 minutes. Let's see. I will start on this, and we won't get all the way through it, but then we'll continue for this next time, right? So hopefully we'll have a hang of what's going on. But these were manipulation experiments. And so what I'm going to show here, there's a little key down at the bottom that indicates what these different symbols mean. The black symbols indicate that this is the natural condition where in this case, the females have an intact ovary inside of them. And we know it's an ovary because I put the female symbol in there. When we come onto the male symbol will represent intact testes. In these experiments, what they did was they removed the ovaries or the testes that are producing the sex hormones in these, and then manipulated them, either took them out but nevertheless gave injections of testosterone, the male hormone that would have been produced by the testes, or gave injections of female sex hormones that would have been produced by the ovaries. So here what they've done is, in the juvenile state, these females had their ovaries, but then they were removed as adults, but given injections of female, and they show the typical sexual behavior, the lordosis in them. In this one, the juvenile female had its ovary, but it was removed as an adult, and then they were injected with testosterone, and nothing happens. They don't mate, they don't undergo lordosis or anything like that. they don't do anything. Okay, that's the start off of that experiment. Okay, and I will continue with that experiment. Don't worry by the fact that we're behind. Some chapters are longer than other, so don't get concerned about that. We'll continue with this experiment in the next lecture. Okay. Office hours in half an hour, start between 2 and 3. There's the link. If you go on to Blackboard, there's a link for that if you need a Zoom link. Okay. Thank you, thank you, thank you, and thank you all as well. Have a good day and have a great weekend when it's supposed to get warmer than it is now. Okay. Cheers then.